DNA replication. Introduction. Watson and Crick, while suggesting their model of DNA structure, postulated a mechanism for its exact duplication or replication. This replication and separation of the molecules occurs during the interphase stage of the cell cycle. The double helix separates into its two constituent polynucleotide strands by breaking the hydrogen bonds that hold the bases together in pairs. The separated single strands serve as models or templates for the synthesis of a new strand. When the process is complete, two daughter double helices are formed, which are identical to each other as well as to the parent DNA. DNA replication takes place within the nucleus. DNA replication is initiated at a region on a chromosome. DNA replication starts with the enzyme helicase acting upon the double-stranded DNA by unwinding the double-stranded DNA and breaking the hydrogen bonds between the base pairs. Helicase unwinds the DNA double helix and the strands separate as the hydrogen bonds between base pairs are broken. Two replication forks form and proceed to separate the strands in both directions. The two strands formed are called the leading strand and the lagging strand. An enzyme called primase then initiates DNA replication by the creation of RNA primers on the leading and lagging strands. Primase synthesizes the RNA primers. There is only one primer on the leading strand. There are multiple primers on the lagging strand. Note the uracil bases in the RNA primers. The enzyme that acts on the two strands is DNA polymerase, which begins to synthesize the rest of the DNA on the two strands in a 5-to-3 direction. DNA polymerase synthesizes the new strand of DNA contiguously on the leading strand and discontiguously on the lagging strand, producing short Okazaki fragments. It is now the turn of another DNA polymerase to act upon the newly replicated DNA to remove the RNA primers and replace them with DNA. Notice that uracil bases in the RNA primers are being replaced by thymine bases in the newly synthesized strands. Now, the enzyme ligase steps in to complete the broken sugar phosphate backbones at the two ends of the Okazaki fragments. Notice that the gaps in the sugar phosphate backbone are sealed when ligase forms the lacking phosphate ester bond. Now, there are two identical double helices of DNA. Thank you.